This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 48 for September 4th, 2009. Hey everyone, it's Vincent Racaniello. It's time to talk about viruses. I got a great panel today, starting with Dick Despommier.
Hey, Vince.
How's it going, Dick?
Going well.
Fishing?
Yep, just recently.
Excellent. So that means you're in a good mood.
Absolutely.
We have Alan Dove returning.
Yep, here I am.
Missed you last week.
Yeah, sorry I couldn't make it.
It's okay. You're making a living. It's okay. We don't, and you do. No problem. You have good weather there, I presume?
Yeah, it's quite unusually pleasant here in
Nice
western
day.
Massachusetts.
And then our final panelist has probably the best weather of all. Rich Condit returns. Hey, Rich.
Great to be here. Hi.
Last time I talked, you were in Oregon?
That's correct. In the high desert just east of the Cascades.
Right.
And are you happy to be home?
Yeah, I'm happy to be home. Back in Florida, home of the Fighting Gators.
Are you a football fan?
Well, sort of. I mean, you know, Dick Moyer and I have season tickets, and we go to a fair number of the games. But you know, I'm not a rabid fan.
You're ranked number one right now.
Listen, this is going to be quite a season. They're really trying to do something pretty amazing, and I think they got a chance to do it.
I would agree with you.
Well, I was on a study section in June, and one of your faculty colleagues was next to me. I don't remember her name, but she had a big Gator sticker on her laptop.
Yeah, Mavis McKenna.
Exactly.
Yeah.
I guess she was a fan or is a fan.
Yeah. Well, you can't live here and not at least be aware of what's going on.
This is all true.
Well, I actually went to a football game this week, preseason game, which is very unusual, but it was fun. Okay, let's talk about viruses, because people don't tune into TWiV to hear about football, that's for sure. No. And the first thing I would like to do is have you, Rich, correct our error from TWiV 47. We're talking about a vaccinia infection in a lab.
Right.
And so fix it for us, Rich.
Okay, so really the essence of the error was the notion that treatment of a vaccinia infection with acyclovir will be actually effective in treating the infection. And vaccinia is resistant to acyclovir. It is true. So acyclovir, as you pointed out, its effectiveness is dependent on the virus encoding a thymidine kinase, because acyclovir is a nucleoside analog. It looks like one of the precursors that goes into DNA, but it's not active on most cells because although they have a thymidine kinase, which activates, shall we say, by phosphorylation, that nucleoside precursor, the cellular thymidine kinase can't recognize acyclovir and phosphorylate it, so it's inactivated in uninfected cells. In herpes-infected cells, herpes virus carries a thymidine kinase that has a really unusual specificity in that it can recognize acyclovir and phosphorylate it. So if there's a herpes virus in the cells and therefore the herpes virus are trashed by the phosphorylated compound, which gets incorporated into DNA and causes chain termination, and also inhibits the DNA polymerase. The vaccinia also has a thymidine kinase, but it does not recognize acyclovir. So vaccinia is resistant to acyclovir.
Do we know why it's resistant, or it doesn't phosphorylate acyclovir?
Doesn't phosphorylate acyclovir. Does not recognize acyclovir as a substrate. So it's just a matter of substrate specificity, and the herpes thymidine kinase is peculiar in that regard, in recognizing acyclovir.
Has anybody fine-tuned this? I'm sorry to interrupt, Alan. You had a comment.
I was just going to say, you could say herpes has a more promiscuous enzyme, perhaps?
Yeah, that's how I would say it. That's correct.
And nobody's tweaked the acyclovir to force it to be recognized by the other thymidine kinases, I take it.
No. And first of all, of course, you don't want it to be recognized by the cellular
that's true.
Thymidine kinase.
Yep.
And in the case of vaccinia, the focus has been on other antivirals. There are actually 2 drugs out there that will inhibit poxvirus infections. One is cidofovir, which is another nucleoside analog, which has recently been modified to, there's a lipophilic derivative that works better than the normal stuff. And another is a new drug that's still in clinical trials called ST246 that holds a lot of promise against most poxviruses.
Right.
So, in the original article that I had seen, which is I think in Reuters, it said an infectious disease specialist diagnosed the vaccinia infection and an 800 milligram dose of acyclovir was administered. So that must be incorrect, right?
That is incorrect. If you look at the original, there were a couple of mistakes in that Reuters article that are worth pointing out. The first is that one. If you look at the original article, which was in the late July issue of MMWR from the CDC, there was a period of time when they had determined that this infection was probably a viral infection, not a bacterial infection, but they didn't yet know it was vaccinia. And during that period of time, they administered acyclovir.
I see.
Okay, so they knew the guy had a virus infection. They didn't know it was vaccinia, so that they used the most common antiviral to cover actually an exanthemus disease. It was a blistering disease, okay? And herpes could do that, even though it didn't look like a herpes infection, really. So they did not do an inappropriate thing. The other issue with the Reuters article was it says here that CDC has changed their vaccinia laboratory procedures. That's also inaccurate. They say that they've changed them to counseling and education plus offering smallpox vaccine to all lab workers with any possibility of occupational exposure to the attenuated virus has been added to the CDC's policy. In fact, CDC's policy from the get-go has always been counseling and education and vaccination. At this institution where this happened, they were not doing. I don't know if they were doing any counseling and education. They weren't offering vaccination, and now they beefed that up a little bit. But they still don't require vaccination at that institution. There's no law that says you have to do it. It's NIH and CDC's guidelines. But
right. We
vaccinate people here.
It's required?
It is required here, yes.
Yeah, here it's recommended. It's not required for working with vaccinia. So what do you think happened? Do you think he got some in his eye, basically?
Actually, if you look at the lesions, the primary site of infection is his earlobe. And I actually did a little looking into this even before we communicated about it because it came across my desk and it was interesting. He had an earring.
I was just about to suggest
that he played with. Okay,
just
and
about to
vaccinia likes to get into, or it infects when it can get into an open wound or lesion, any sort of break in the skin, put it that way. That's how it infects. And so he probably had some on a glove or something like that and tweaked his earring and boom. And he had numerous secondary lesions, including the most serious in his eye. I assume that was a secondary lesion.
Right.
And that's probably from getting virus from the primary lesion on your hands and spreading it by contact elsewhere.
Thank God he didn't have a tongue bolt.
Right.
Or
no, they don't go there. Don't go there.
Any sort of mucous membrane is susceptible. So there you go.
That's great. All right. Well, it just goes to show that gloves are not going to help if you touch yourself with contaminated gloves, right?
This is not the first time I've seen that happen. Somebody with a contaminated glove wipe their eye or something like that. Boom. Yeah. It's common sense is the bottom line.
Tell me all about it. The funniest thing I've ever seen is the misuse of protective garments and gloves in delis, where the guy is standing there for 2 hours making sandwiches with the same pair of gloves. He's actually protecting himself, not you.
Yeah. That is great that you caught that. Now, I understand, Rich, that you listened to TWiV on your bicycle, so you probably fell off when you heard this error of ours, right?
Right.
Can you believe this, guys? He has a Bluetooth earphone, and he listens wirelessly to TWiV. I think that's great.
You don't put it in both ears, do you, Rich?
Listen, you raise a very important point because it's actually illegal to ride a bicycle with a headphone on both ears, right?
No kidding.
I have been busted by the campus cops for this.
Why?
Didn't know that.
Why?
Because you can't hear traffic coming.
What if you're born deaf?
Well, listen, I could have this argument
not
with the
win,
cops, but it's not going to do
So
me.
one ear is okay.
Exactly, which is why I got the Bluetooth device.
Oh
Plus,
my
it's cool.
gosh.
You're very cool,
Plus,
Rich.
that's right.
That's great. I'm glad you listened to TWiV. All right, you going to listen to yourself now?
You bet.
Yeah, sure.
Absolutely.
I end up doing that just to make sure we get everything right. Okay, so that's one error that we fixed from TWiV 47. And then the next item I wanted to talk about is a continuation of last week's theme, which was the vertical vaccine farm. And Russell sent a link to a story called Tobacco Plants Yield the First Vaccine for the Dreaded Cruise Ship
Virus.
I think you also
Yes, I did.
looked at this one too, Alan. So last week, Dick and I talked about making virus-like particle vaccines in plant cells. It turns out there's quite a effort to do this. For influenza, anyway, you can just express the hemagglutinin and you get particles made. And I'm going to give you some links in a moment about the companies that do this. But this... in this case, it's a vaccine against noroviruses. They're making empty particles.
Which is named after a small town in Ohio, as I recall.
Not Connecticut.
No.
That's right.
No.
Yeah. And these are, of course, the major cause of gastroenteritis and often outbreaks on cruise ships.
Not a good thing to get.
Food handlers.
Yeah.
Very unpleasant.
That's right.
So they've made these nanoparticles in tobacco plants.
Cool.
And this is fabulous. So I have an article here from Eureka Alert. I think that's what Russell and Alan had. And then there's also an original research article in, what's the journal here? Vaccine. It was published in March 2008. An efficient plant viral expression system generating orally immunogenic norovirus-like particles. So the idea would be you take these orally. They're not infectious. And they immunize you. So they're at the stage of putting these in mice. And they have to give you quite a bit, 100 or 250 micrograms. And I suppose that's because you're taking it orally.
Right.
And a typical flu shot is 15 micrograms. It's going right into your muscle there.
But norovirus would be ideal for that, taking
Perfect.
early.
Yes, of course, the intestine is the site of action of the virus. That's
Sure.
where you want the intestinal immunity, right?
Exactly.
So that's perfect. Now, so now we have norovirus vaccines and influenza vaccines. And I had blogged about this at Virology blog a while ago, and someone left a comment. He said, a company named Medicago, and it's at medicago.com, is doing this technology with tobacco plants. And he says 1 square meter of plants can produce 30,000 doses of vaccine at under 20 cents a dose. Shows full protection at low doses, and for H5N1 influenza cross protection. And he sent me a PowerPoint presentation, which is for investors, but I'm going to put that up on TWiV.tv because it's nice to look at as a great summary of this whole process. Now, Ed Rybicki, who is a virologist in South Africa, apparently has done some work with expressing... Do you know Ed, Rich?
No.
So he's expressed some virus proteins in plant cells. So he wrote at my blog, suffice it to say that conventional vaccine production requires large amounts of stainless steel and sterile manufacturing conditions just to produce the raw material. And production of the raw material for a plant-produced vaccine does not.
Correct.
My lab has made equivalent products, virus-like particles composed of human papillomavirus L1 proteins via baculovirus insect cell culture and via transgenic and transient expression in plants. And I can vouch for it costing a lot less for the infrastructure to produce the plants than it did for all the insect cell culture facilities and media. Simply harvesting virus-like particles from culture medium requires filtration, centrifuges, possibly columns, which is exactly what the downstream processing for a plant-produced vaccine would need. And he goes on to say, interesting thing about the Medicago HA proteins and the ones we have experimented with, they localize to the apoplastic space in leaves, which can be flushed out in intact leaves, infused with simple buffer by vacuum infiltration, then low-speed centrifugation without damaging the leaves. You end up with straw-colored liquid rich in HA protein with very few contaminants, pretty much equivalent to harvesting tissue culture supernatant. So basically he's saying that you can infuse the plants with buffer and then spin the leaves down to get the buffer out, and it washes out the particles.
Well, and if the tobacco leaves
great.
are still intact, then you could roll those up into cigars, right?
That's right. That's right. Hey, Alan, do you remember at a Gordon conference years ago, they were talking about this, and at the break when everyone went outside to smoke, we joked that they were going to smoke the vaccines.
Yes.
Remember that? Yeah, I was thinking of that. Well, Dick, you mentioned last week they're doing this with hepatitis B in bananas.
That's right. It's
So
an older technology to express the surface coat proteins and to have them ingested by small children to protect them against hepatitis.
Well, so in the case of norovirus, you could do that in a banana and just eat the banana.
And polio. I mean, I don't know why you couldn't do it for polio.
But of course, that doesn't. You don't have a controlled amount of antigen, I would guess.
No,
so it's
you're right
better
on
to
that. I think that's the
purify it.
danger is to elicit either tolerance or some weird thing might happen that you might not express this immunity in the right way.
So
right.
anyway,
You need to get the immune system to recognize it not only as foreign but as a threat.
That's right. That's right.
So anyway, this is very exciting. And there's another company just out of completeness, Fraunhofer in Germany, which is also involved in plant-based vaccine production. There's quite a bit of literature on this, and I wouldn't doubt that this is going somewhere at some point.
Sure.
It's very cool.
Plus, you don't have to worry about retroviruses from animal cells and that sort of thing getting in the way of things. And I think it's a very clean technology, actually.
Well, there is still some controversy about if you're growing these in open fields.
That's different. No, it's different.
Yeah, if you're using depending on the plant you're using, if you're using a nightshade like tobacco and it transfers its DNA to local tomato plants,
Yeah, exactly.
then what are you doing?
That's right.
Which would be another argument for growing them indoors.
You bet.
And that's where it comes in the vertical vaccine farm.
Yeah.
Yes.
That's right.
So, Dick, you thought you were going to do food, but now you're going to do vaccines in your vertical farms.
Apparently.
Great. Okay. Next is a story found by Alan.
Yes. I don't have an iPhone, but obviously it's very much in the news, and I saw this story about a new iPhone application called Outbreaks Near Me.
Is that like Toys R Us?
Kind of like that. So you can look up names and addresses of friends. You can find all sorts of interesting things apparently on your iPhone. And now you can find news about outbreaks that are in your general vicinity.
Wow.
So I was listening to you guys this morning from a November podcast on my bike, on my iPhone.
Oh, dear.
And neither of you guys had iPhones either, Dick and
No,
Vince. Is that
I
correct?
now have a BlackBerry, but I don't know how to use it.
So you have an iPhone, Rich?
You bet, and I downloaded this thing last night.
Tell us
Really?
about it.
Oh, it's terrific.
Is it?
So you boot this thing, and you get a map.
Yeah.
And these iPhone apps, any iPhone app that has a map function is integrated with Google Maps.
Oh, right.
Okay? And they'll either allow you to search for an address or it will by default find where you are, okay? Because it's a GPS
yes.
too,
Oh, yes.
okay? So if I just log into this thing, it shows me it seems to do it by state. So if I look for my current location, it starts out with a map of the world and then zooms in and drops a whole load of pins on Florida. Okay, so I got a Google map here, and it's got a green pin on Gainesville showing where I am, and a slug of red pins.
Cool.
And if I hit any one of those red pins, it shows me why it's there. And what this thing does is, you're probably going to describe this, Alan, culls public information, newspaper stories, ProMed, etc.
Wow.
Right. Essentially, so what it's doing is it's I think the Health Map website might actually have been a pick of the week some time ago. So it's essentially a front end for the iPhone for the Health Map website. So it, as Rich just said, it maps your location based on where the phone is, and then plots it finds all the stories that are related to that in all the public resources, whether they're wire stories or I think it may check ProMed as well.
Yes.
And it checks all these resources and looks for, West Nile virus plus Florida or influenza plus Florida, and then it'll give you the stories and where they're located on the map.
That's outstanding.
Yeah,
the other aspect of it is they've built in this, and Rich, I hope you haven't had an application for this yet, but maybe you will. There's apparently the option to submit an outbreak report.
That's correct.
Right. So if you believe that there's an outbreak of something in your vicinity, anybody with an iPhone can pick it up and say, okay, I think there's an outbreak of this disease, and here's my location, and you can upload a related photo, and I presume that becomes part of the health map feed.
Yeah, what I don't know is whether or not there's anybody to sort of screen that stuff.
Yeah, right.
That is my biggest concern with this.
That's right.
Yeah, exactly. Right.
Because if there's nobody riding herd on it, I mean, we're going to see anthrax cropping up all over the place, and just.
No, you can imagine people are going to fool with it. Sure.
Yeah.
So there's another feature here, and I don't know if this is on the regular version. It must be. There's an alert function.
Mm-hmm.
So I can. There's a feature that's alert settings. So I can have this thing alert me.
Got it.
Anytime by email, anytime there's an outbreak somewhere, and there's a list of things that I can check that I'm interested in. You know, dengue, chicken pox, cat scratch fever, you name it. It's got a pretty long list here.
No, this is very cool. This was made at Children's Hospital and MIT. Very
Really?
cool. Yeah, researchers at Children's Hospital Boston in collaboration with the MIT Media Lab.
Oh, yeah.
Who I guess
Famous place. It's a very famous place.
Yeah, this is very cool. I just don't know about the reporting and if someone's going to, as you guys said, vet that, because that could be a problem. But just seeing what's going on. So what's going on around Gainesville?
Well, I'm glad you asked, because we look pretty clean right now.
Oh, that's good.
The first thing, the closest thing I can see is down near Orlando. There's 2 pins here. One is, let's see here. This is cancel practices following a flu outbreak at some high school. That's one of them.
Oh, yeah.
And the other one is in Altamont Springs. Oh, that's the same thing. Lake Brantley cancels practices following a flu outbreak. Most of these are swine flu.
Sure.
But I saw something with a dog intestinal disease or something like that.
The next week is going to start heating up. As kids go back to school, we're going to see a lot of influenza, I bet.
Right.
Yeah, because it's been around all summer. It hasn't gone away. And as soon as these kids get back together again, boom. So we will see. But that's great. I wish I had an iPhone.
Hey, you could
Verizon,
buy one.
You know, Verizon.
That's right.
I'm waiting for Verizon to get it. I hear it's coming. Come on, guys. We need this for TWiV.
Yeah.
Well, I've got a BlackBerry. I'm waiting. I
guess these guys could write apps for Blackberries, too.
Sure.
Well, there are other platforms. There's some other phones that you can write apps for, whose names are escaping me. I think the G2 by Google.
Yeah, Google is supposed to have theirs fairly open.
And I think this involves some Google business, too, the searching, right, the health maps. Or that's another.
Yeah.
So
all of these map functions, at least on the iPhone, integrate with Google Maps.
So I wouldn't be surprised if this pops up on the G2, too. Okay, that's great. All right. Now, before we go on to a couple more stories, we have a word from our sponsors of this episode, Citrix, the makers of GoToMyPC. If you're tired of spending late nights in the office, it's time you tried GoToMyPC, because with GoToMyPC, brought to you by Citrix, you can access your office computer from home or anywhere with an internet connection. So you can leave the office early, have fun, and finish later when it's convenient for you. Dick is laughing. He doesn't have fun. It's very simple to do. You go to their website from whatever computer you want to share. You log in. You download a little piece of software. You'll have a login ID and a password, and then you can go to any computer with a internet connection, go to their website, put in your username and password, and there you will have your desktop from your computer, and you can access all your files. You can run programs, email, and access your network resources as well. Absolutely easy to do. And you can try GoToMyPC free now for 30 days. Visit gotomypc.com/podcast. That's gotomypc.com/podcast. You'll get a free 30-day trial. We thank them for their support.
So, Vince, they also do a conference call setup, too.
Yeah, GoToMeeting, I think it's
That's
called.
right, exactly right.
Yeah, I've used that. It's very nice. And when TWiV is at 1,000 employees, we'll have to get that.
I think we might. That's right. We better anticipate this one.
Actually, at 10 employees, we'll get
3
it.
would be fine. Sorry, Alan. 3 in-house employees. How's that?
Right. Well, no, employees suggest that there are checks involved.
This is all true.
Yeah,
One employee, for that matter.
Now we know what it's like to be an intern.
When we start getting paid, we'll do that.
That's right.
All right, I just wanted to mention this story. Norovirus fears spark flying fox cull calls, which we actually talked about
Oh,
a
dear.
couple weeks
Sounds
ago.
like the start of a Dr. Seuss.
Yeah, right.
Yeah, it is a
Exactly.
tough title. This is from news.com.au. And a couple of weeks ago, we talked about the idea Hendra is back in Australia. It's killed horses and I think one horse veterinarian so far. And there's a call to kill the flying foxes, to cull them, because that is probably the reservoir or one reservoir of the virus. And we discussed this previously, and I just wanted to bring up this article because the government has rejected calls to kill the flying foxes.
Good.
And a Wildlife Queensland spokesman said it's not even clear if they're the root cause of the disease. We would like to see more positive advances in finding the true cause of Hendra and its route of transmission to horses. Calling for culling of flying foxes is a pointless retribution. Wiping out a native species that is part of our natural biodiversity doesn't solve the issue.
Here, here.
Good point.
Here, here.
Good. Yeah, that's
And
great.
we all know that horses are not native to Australia either, so perhaps they could just be moved away from the endemic zones where flying foxes live.
That's tough.
Yeah, I know, because that's
a
where all the
tough one.
racehorses...
As you said, they bring the racehorses from other countries
They do.
as well. So
They do.
that's a tough one. Okay, the next story is a find again of Alan's. Needle-free inhalant powder measles vaccine.
Oh, can I interrupt just for one moment?
You can.
Have you heard about the civil unrest that's going on in upper western China right now? There's a city in which there was a series of, I guess, aggressive acts by some presumably crazy people, but they've carried syringes with needles and they go around sticking people with them.
What's in the needles?
It hasn't come out in the news reports yet, but China has declared a state of emergency for this one city, and they've gone in and people are starting to react against martial law being instituted and curfews and this sort of thing. But it all started with people running through the streets, stabbing other people with syringes with needles on them.
Well, it started before that.
Do you know
with-
more? Do you know the origins for that, Alan?
Yeah, I think that's the same city where they had these riots and uprisings of there's a large Uyghur population there.
Mm-hmm.
I think that's the same place, and so there's been a lot of unrest and.
What's the needles and syringe thing? What's that all
Well,
about?
that's just the latest phase of it. I think before it might have been explosives and knives, and
I
now
see.
apparently people are running around with syringes. I don't know. Maybe the explosives and knives got confiscated, but so there is a background of unrest going on, and it has to do with this ethnic group that feels perhaps correctly that they've been oppressed.
Right.
That's my understanding. It's my somewhat sketchy understanding of it, but
All right.
yeah, there's a little.
I know
bit
there
of
was more to it than I was saying, but
Yeah,
Well,
but it is odd that someone chose to go around with needles
exactly.
as a weapon.
Probably, I would guess they're empty.
Yeah, I would too.
We'll
but
find out. If they're viruses, we'll let you know on TWiV.
Yeah,
but it does tend to create a phobia against being injected.
yeah.
Well, this vaccine here may get around that. Right,
See,
Alan?
that's why I mentioned
That's
it.
a segue, Dick.
That was
That's
a
right.
segue. That was
Thank
a
you very
segue.
much. I don't ride one myself, but thank you.
No, well, they're actually
So
quite
yeah, this
interesting.
is a another story that came out of the American Chemical Society meeting, which I guess was a week or so ago.
Yeah.
And this is an inhalable measles vaccine. Now, as I'm sure TWiV listeners are well aware, delivering a vaccine is one of the ongoing problems. You've got to line people up and give them shots, and in developing countries, it's hard to get enough needles and keep everything sterile. So the inhaled vaccine has been kind of something that people have been interested in developing for a long time. And these folks, the lead investigator is Robert Sievers. He's at University of Colorado Boulder. He was originally an atmospheric scientist, so he was approaching this as from a physics perspective, and he and his colleagues came up with this method that uses supercritical carbon dioxide. So I guess it's partially liquefied, or it's liquefied CO2, and that is, I guess, shot at the virus prep, and it produces these microscopic bubbles that they dry and turn that into an inhalable powder. And then they've got there's a photo with the press release of this a woman with the collapsible bag that she's inhaling from. So they're working on the delivery system. And they've tested it in animals. They've found that it produces an immune response and appears to work. And now they're working up to set up human clinical trials. So this would measles is a great target for this because it's an injected vaccine and it's something that obviously we'd like to be able to wipe out, and so that's kind of where they're going with it initially. But it's a technology that could, in principle, be applied to just about any virus, I would think.
This is a good one here because measles is a respiratory infection,
right?
And so this is infecting right where the virus usually goes, and that's more natural than injecting measles vaccine, of course, even though, there is a viremic phase, so injection works, but this would be theoretically even better.
Right.
Now,
so what puzzled me about this is that measles is an envelope virus, and apparently this is the attenuated virus that they're using, so it's supposed to work through replication, and sort of the rap that I give the medical students that we all live by is that enveloped viruses are sensitive to drying.
Yes.
Okay. So if you dry them, you'll kill them. So that seems inconsistent with this. Obviously, this works in animals, so something's going on.
Is this infectious, Alan?
I don't know. That's not been spelled out. And this was delivered at a meeting. I don't have a full report of it. It'll be interesting when the paper actually comes out to see if they've looked for replication. Hopefully they have. Or whether they're just delivering antigen. And for that matter, if just delivering antigen this way is enough, then maybe we don't need to bother with the whole virus.
I have an article here, a new rapid and promising approach to aerosol immunization, inflatable bags, and it's from a different group. It's from Mexico, and they use an infectious aerosolized preparation, so it's not dried with this method of CO2. They use it because they say typical inhalers waste a lot of material. So if you put it in these bags, it's a little more efficient. But in their case, it was infectious because it's just aerosolized. It's not changed in any way. So I don't know this needle-free inhaler. We'll have to look that up. I don't have any information here.
Yeah.
I've got a question, and that is just a random thought that just occurred to me. And of course, I get lots of them just sitting here looking out the window. Has anybody ever tried to deliver a vaccine in chewing gum?
I'm not aware of any.
Because there's a special chewing gum that you can buy with a liquid center.
Yeah.
It's got something like a flavor burst or something like this. But imagine, for kids, and it's a single dose, and it's got 3 or 4 different viruses inside. The Norovirus, and of course they've got the poliovirus, and
why not?
And rotavirus. And
why would that be? Why would that be easier than a liquid?
Well, because it's a slow release thing, and kids love chewing gum, and you make it sweet, and you give it to them, and they make sure to keep it. It will actually improve compliance, perhaps. Okay. And the other thing I thought about was that if you're chewing gum and you're inhaling at the same time, there's an aerosolization of the material in your mouth, and so this measles virus might fall into that category of being able to, creating an aerosol inside your mouth, and every time you inhale, of course, you're back flushing whatever in your oral cavity down into your lungs, and maybe that's another way to deliver this too. I don't know, just a thought. Well,
you better patent it, Dick. Hurry up.
No, I threw that out for the general consumption.
Okay, open access.
That's right.
Idea. So here they have this interesting. The powder is puffed into a small sack with an opening and administered. You take one deep breath, and the child's vaccinated. So I guess that's better than inoculation. And the question is whether this is infectious. I can't tell. We'll have to
have a question for you. What do you do with the plastic bag after you finish with it?
Hey, it's biohazardous waste, right? If it's infectious, I presume.
Right, so you couldn't give this to somebody and tell them to take it home and use it. It has to be done in the eyes. So. It has to be witnessed.
Yes, I would think so. Yeah, but they do say this is a perfect fit for use in backroads areas of developing country where they don't have refrigeration, clean water, and sterile needles. Right. So that's good.
And when you say it's biohazardous waste, I mean this is an attenuated vaccine, and you are delivering it to people anyway. So it's not. It's not as if a child picking it up and playing with it would be in danger. They'd just be vaccinated.
But it's still, you would agree that you don't want to.
Oh no, you don't want to. Yeah, you don't want to throw these out. You would pile them up in the corner and then.
And they'll all end up in the Western Pacific Gyre.
Yes, right. No, you want to bury them underground, I think.
Yeah. Well, Rich, your point is well taken. Is this infectious or not? Because we all suspect that drying an envelope virus would be bad.
Right.
And I've heard that this is why a lot of enveloped viruses cannot be weaponized, because they have to be dried in order to be weaponized, and that kills the.
The other possibility is that this rather unusual method that they've used has maybe cracked that problem.
Perhaps. Well, we'll look it up and let you know, but maybe in the meantime someone will email us, which often happens.
That would be great.
Because our readers, our listeners are on the ball here. They know what they're doing. All right. I would like to move on to email now, because we have a few, and they're very interesting as usual. The first one is from Juliet, who happens to be a former PhD student of mine. Just finished a few months ago. Hi, Vincent and Dick. Great job on the netcast. I started listening to it last week. Can you believe it? Just last week?
Your own students.
Yeah. To brush up on my influenza knowledge, and now I am absolutely hooked. The past episode you discussed viruses that may confer some advantage to the host. It made me think of the paper from Skip Virgin's group at WashU. Herpes virus latency confers symbiotic protection from bacterial infection. That showed that in mice, at least, a chronic infection with herpes virus prevents infection with listeria and Yersinia. That was a pretty thought-provoking piece. I also really like the Virology 101 idea, as it allows me, a new postdoc in a virology lab, to brush up on the field without reading a textbook. Keep up the good work, guys. So, Dick, see, it's good to do Virology 101.
Oh, absolutely.
So this is a very interesting paper. I don't know if you guys are aware of this, but they infect mice with murine herpes virus, gamma herpes virus 68, or murine cytomegalovirus, which are similar to the human viruses Epstein-Barr or cytomegalovirus. And these mice become resistant to bacteria, listeria and Yersinia. And the mechanism is that the virus, which becomes latent, upregulates interferon, and that provides protection against infection. So it could be that, these latent lifelong infections actually confer some protection. Now, of course, this is mice, and we don't know if this is this case in people. But
yeah, I just recently read an article where a parasitoid wasp injecting their larvae into a host caterpillar. Some of the caterpillars have a virus inside them that actually kills the parasitoid egg. And then protects the butterfly, which is going to happen eventually.
Because some of the wasps put in a virus that...
That's right.
It suppresses the immune response of the...
It's working in both directions here. By the
Very
way.
cool. Well, in fact, one of the papers that we didn't have time to do is similar, having to do with aphids, which we'll do another time. But yeah there are all kinds of... Excuse me.
Vincent just sneezed, in case anybody wondered what that was.
All kinds of...
that was not a 7.2
There are more of these symbiotic relationships coming up. It's very interesting.
It's quite amazing, actually. It really
I think that herpes paper is just terrific. That
beautiful.
Opens up all new doorways. It's very interesting.
I have another question. Of course, these are all random questions, but and they can be edited out. Vince, go ahead. There's a bacterial species called Wolbachia, and it's everywhere. It's almost in everything. It's hard to get rid of this darn thing. And I just wondered if Wolbachia has a phage.
Sure. I bet it does. I would bet you $100 it has a phage, Dick.
You would,
yeah, most bacteria have phages. Sure.
Well, I would agree with you in that sense, but I wondered if someone out there could help
Why
us.
are you thinking about a phage in Wolbachia?
Well, because that's yet another level of regulation for whether or not something that's harboring Wolbachia could also, be affected by the virus inside the bacteria, which then affects the organism that it lives inside of.
Yeah.
So it's got layer upon layer upon layer.
Yes, so there is a bacteriophage called WO.
WO. Look at how fast that internet info is.
In Wolbachia.
That's incredible.
You knew I didn't know that off the top of my head.
Well, I'm glad I asked the question then. How's that?
Yes.
All right. Andrew writes, he sent us a link to an article called Mutant Poliovirus Spreads in Nigeria. It's from a Detroit radio program. And he asks, how bad is this information? How badly did the AP screw up the story? Is it true? Can you guys address this on your next episode? So actually, we did this a few weeks ago, right, guys? We talked about type 2 polio reemerging in Nigeria. It's vaccine-derived, in fact, and Nigeria, of course, has a checkered immunization history. And I think the article is pretty good. What do you think,
Yeah,
Alan?
I thought so. I couldn't. I thought they did a pretty good job. Most of my information, of course, comes from listening to your broadcast on TWiV a little while ago. So that may be messed up,
It
but
could be.
certainly the article was consistent with that.
We're not perfect. Yeah, we're
this
not
is
perfect.
this is certainly consistent even with what the WHO has been reporting on this, and this is what's happening. It's a vaccine-derived virus that's now circulating in the human population, and this is a well, not to brag too much, but this is a predicted consequence of doing this type of campaign.
So it's we talked about this in TWiV 45, Andrew, so check that out. You probably have already heard it. This is a couple of weeks old, this email. I'm sorry we are so lagged in getting to these, but yeah, it's a good story. Okay, someone named Rich Condit wrote in this episode, which again was a while ago, you asked, what would you say if you had the whole world's attention for 5 minutes? Wow. Great question. Hey, Rich, that's you.
Oh.
Why don't you just read your answer?
Okay, so I said, here's my answer. I would first quote the Buddha. Quote, believe nothing, no matter where you read it or who said it, no matter if I have said it, unless it agrees with your own reason and your own common sense, end quote. Then I would give a very short description of the scientific method as a good example of how to test whether anything agrees with your reason and common sense, and I think there would be time left over for questions.
Yes.
Right.
It's nice.
It's brilliant.
So,
it's good. I like that.
If you had the world's attention for 5 minutes, you would teach them how to think.
Exactly. Teach them how to learn.
That's right.
Yeah.
The problem with it is that once they run into a conflict with their own belief systems, they tend to create excuses for why you're wrong and they're right.
Then their belief system becomes common sense. Yes,
that's exactly.
This is really interesting because I had an opportunity to give one of these sort of touchy-feely talks at a graduation a while ago, and I talked about the scientific method as part of this talk and about how it was important to me as a way of looking at things. And I said, okay, so what's the scientific method? You create a hypothesis, you construct experiments to test your hypothesis. If the experiments are inconsistent with the hypothesis, you change the hypothesis to conform with the data, and then you repeat the process. And when I said that, the audience burst out laughing. The notion that you would change your hypothesis to be consistent with the data was foreign to them. Okay? And that surprised me. And I thought, whoa, this is a wake-up call. What are you going to do? Change the data?
That's right.
All right?
That's
or just ignore the data or something? Yeah.
Those are all common responses, yes.
It's very important if this process is going to work that the ego be dissociated from the process.
Here.
The ego compromises the whole process.
Yep.
Yeah, great point. That's all good stuff. I like your 5-minute thing, and this little discussion is really important. A lot of scientists, never get into this, and they do their experiments, and they say this is the answer, and that's it. And they don't understand that non-scientists and scientists have some other issues dealing with their own reason and common sense. So that's good. Now you
have to take just as much pleasure in being wrong as you do in right, because both of them are pathways to the truth.
Absolutely. Here. All right, so that's good. Now you've done it, and Dick has done the 5-minute thing. So it leaves Alan and I for a future episode to come up with the 5-minute attention thing. But I like that. Thanks very much for that, Rich.
Well, thanks for giving me the opportunity.
Sure. Okay, Barry writes, all right, so a few weeks ago we criticized a podcast called No Agenda because they were talking about swine flu. And Barry writes, I listen to No Agenda because of the pair's banter. Their off-the-wall conspiracy theory stuff is mildly grating to listen to. Their ignorance is blatantly clear, and their anti-government, particularly anti-Obama political stuff, I mostly just skip over. But when they talk about other things, it's amusing. More than anything, listening to their show gets the blood riled up, and myself and my girlfriend can spend a few minutes mocking their ignorance. Okay, thank you, Barry. Not everyone agrees with us. There's some trashing of TWiV going on at the No Agenda boards, but that's life.
That is life.
Okay, Lotem wrote, I found out about you not too long ago, and I'm enjoying every episode. You came in pretty handy on the last CSI LA episode. My husband and I have a contest on who figures out the murders first. The one we saw last night, I set a new record thanks to you guys. It took me 4 minutes to come up with rabies.
Ha!
And ruin the episode for the hubby. LOL. Keep up the good work. Hey, that's pretty cool. Wow. CSI. I don't watch this thing. Do you ever watch this?
No, for me that stands for Construction Specifications Institute.
I see. Okay.
I gave a talk at that last year, by the way. It was a fun-filled, packed audience of 55 people. But think of who they are. This is the group that developed the LEED certification for buildings. So they do a lot of good.
I can see why you'd need to do that, Dick, with vertical farms.
Well, they don't have a category for it yet.
Yeah, they will, and you're going to develop the specifications.
But I'll tell you what's happened recently in New York City, which is quite amazing, actually. There's been a scandal with regards to the making of cement. And the specifications required for cement is rather stringent. And the testing laboratories that were involved in this scandal obviously gave incorrect readouts for the structural integrity of the cement that was being created by these companies. They were skimping on the expensive ingredients and filling it up with other stuff. This is not a once-in-a-lifetime thing. I mean, this happens all the time. So who oversees the process? And New York City has decided to develop its own laboratories for certifying cement as a good material, whether it's got all the right materials inside of it or not. So this sounds like an oversight committee that would be involved in looking at drug manufacturing or in food additives or anything else that goes on that you consume.
No, we have the
Only
FDA,
in this
right?
case, we expect to see some concrete results.
You certainly do.
Don't we have the FDA for that, Dick?
2, 3, 4. Excuse me? We have the FDA for a lot of things, but I can tell you that there was a recent survey done by a scientist at Georgia Tech, which isn't too far from you, Rich, who discovered that 50% of the drugs that are bought over the internet as generic drugs don't have the drug inside the vial. And that's really tragic. 50%. That's a very high percentage. So if you're a diabetic and you order insulin and they send you ersatz insulin, you can really get into trouble fast.
This week in CSI.
Yeah.
No, this week in concrete. TWiC. Okay. Lisa Ann wrote, "Hello to the TWiV hosts. In one of your past episodes, you briefly discussed the reasons why it might not be feasible to immunize large herds of pigs on farms. It's amazing how quickly these issues then appear to be addressed in the latest research. If you have not seen this, enjoy. I found the last line about research location being in Manhattan versus Kansas particularly humorous." This is from Lisa, who's an RN who's written to us before. She sends an article called K-State Lab Gives Researchers the Tools to Study Porcine Circovirus-Associated Diseases. And this is all about a disease of pigs caused by circos, which are little DNA viruses, single-stranded DNA viruses. They're thinking about immunizing, and that's something we've talked about, immunizing pigs for influenza. The sentence she refers to is the existence of the BRI, Biosecurity Research Institute, and the nexus of scientific expertise on animal health and disease at K-State were among the primary reasons the U.S. Department of Homeland Security chose to build the National Bio and Agro Defense Facility in Manhattan. Kansas.
Manhattan, Kansas.
Yes, not
That's
Manhattan.
right. We're sitting here in Manhattan.
You know, they put a very high containment facility in Galveston.
And they put another one in out in Hamilton, Montana.
Well, Hamilton, would be okay, but you know what happens in Galveston, right?
Yeah, I mean, that's just...
They're going to get flooded out.
It's crazy.
Just amazing.
That is nuts.
Did you say pork barrel?
Yes.
The pork barrel virus.
All right. Tom wrote, "Hi, TWiV. I recently got around to listening TWiV 43, which included discussion of Guillain-Barré and the 1976 swine flu vaccine. I'm wondering how it was concluded that the vaccine causes Guillain-Barré." So he sends a link which says that Guillain-Barré afflicts 1 in 100,000 in the general population, just generally all the time. So he says 45 million were vaccinated in 1976, so one would expect 450 cases in that size population. And I had said that there were 500 cases of Guillain-Barré. Can the 500 cases which did occur be explained statistically without considering the vaccine as a cause? For example, there's probably age bias in the vaccinated population, and that alone may explain the extra 50 cases. Or other data used to make the connection, such as a correlation between the time the vaccine was given and the time of vaccine onset.
Yep.
Well, I found a nice article in JID, Journal of Infectious Diseases, and here's the story. I'm not a statistician, but here's what they conclude. The original retrospective study found that vaccine-attributable risk averaged over all age groups in the 6 weeks after vaccination, that was in 1976, was 8.8 cases per million vaccinees with a relative risk of 7.6. The incidence of Guillain-Barré in the unvaccinated populations averaged 0.9 cases per million per month across all age groups. However, subsequent studies indicated that the risk estimates for swine flu ranged from 4.9 to 11.7 cases per 1 million adult vaccinees. So in comparison, the incidence of GBS in the general population is 0.6 to 4 cases per 100,000. So there are some differences only in that year.
Right.
So after that, there is no consistent causal relation between influenza vaccination and GBS. They're just borderline associations. So I'll put a link to that for you so you can see where those numbers come from. It's not a difference between what you would expect and the 500. It's the temporal association.
Exact.
Right. All right. Ivan wrote, "Hello, I found out about your podcast from This Week in Biotech, and I never missed an episode ever since. I'm an electrician in London, UK, and this October I will start an evening university course in biomedicine.
Nice.
Viruses are always been one of my favorite scientific subjects, and I'm thinking to continue in the field of virology after I graduate. In one of your episodes, you were discussing if viruses are alive and that the current definition for life is uncompleted. Can a possible solution to this be the following? Life is any complex system susceptible to evolution. Thank you for this wonderful podcast."
That's an interesting new take on that.
What do you think about that?
Gentlemen.
Well, I've usually defined life as the evolution of nucleic acid sequences over time. That's a much more it's a looser definition than it doesn't include having to have your own metabolism and stuff. So that would include viruses. Okay, I know I've heard Vince talk about this on podcasts before, and he states categorically they're dead. I'm probably biased here. Okay, but I've got this. I think I want them to be alive.
I mean, when you say you've inactivated a virus, doesn't that mean you killed it?
Well, it's
Come on.
It's a semantic problem, Dick. Yeah, I mean,
Well,
we don't. That's not what we actually.
Did you have you had Luis Villarreal on?
We have, yeah.
And what does he say?
I don't think we asked him, did we?
No.
No. Yeah, I mean, I think he waffled.
I'd like to hear his answer.
We have to go back and listen because I don't believe he gave an answer. I don't. This answer of Ivan is very interesting. I've heard scientists use this. It's any system susceptible to evolution. So viruses can evolve once they get into a cell.
Sure.
So they could be living in that sense. But then there are others who say, well, they can't evolve on their own, which is what's required to be living. So therefore,
the other thing is, if you have a definition that's this broad, there are a lot of things that would qualify that you wouldn't normally think of as life.
So what are transposons? Okay.
Well, and what's the computer program called The Game of Life? Have you ever seen this thing? It's a set of computer graphics that evolves over time. It's a complex system susceptible to evolution, certainly, but it's a few lines of code that somebody wrote in the 1970s.
Well, Alan, that's stretching the definition of evolution, isn't it?
Well, it was created by a person, but then it's capable of evolving on its own later.
Yeah, but evolution includes the concept of environment and a response to environment.
It is responding to its own internal environment.
Weak.
Well, as you see, Ivan.
My point is that if you set the definition so broadly, then you start encountering some odd challenges, I think.
Yeah.
Well, Ivan, as you see, there's not an agreement on this, but your definition has been considered. I don't think it. I think it's too broad. But others don't.
However, for a guy interested in electronics that wants to go into virology, that's a damn good start.
It's great.
Yes.
Absolutely. Good job.
Yep.
Now, Swiss Compass and Allison, who is a PhD candidate who's written to us before, both have written to us. Both sent us links to this new game called The Great Flu, which was made by the Erasmus Medical Center in Rotterdam, Netherlands. And it's basically the players try to control a new pandemic from spreading, and you need to pick a flu strain and analyze it for its spread. I haven't played this. Swiss Compass sent a link to a review, which we'll post. And Swiss Compass also said, I initially envisioned an Aidan Racaniello encore on TWiV or an Albert Osterhaus head of virology at the Erasmus Medical Center guest spot, or even both. So my son Aiden reviewed some virus games a long time ago on TWiV. So I asked him. He said he would come and do a review of this one. So we'll get him back. There are actually about 5 or 6 new flu games that have been developed since our first episode on viruses in video games. So I'll get Aiden to review them. And he's a high school student now, so maybe we can pull in some high school listeners.
Sure.
Yeah.
Duncan wrote, "Thanks again for a great show. I thought this might interest you as a crossover between virology and technology." He sent a link to an article called "Single DNA Molecules Self-Assembled in Solution via a Reaction Between a Long Single Strand of Viral DNA and a Mixture of Different Short Synthetic Oligonucleotide Strands on Lithographic Templates Using Traditional Semiconductor Technology." This is according to IBM. He says, "Forget computer viruses. These computers are viruses." It's getting a little blurry, isn't it? Yeah, so as I understand from Alan, you can make DNA into different structures by hybridizing it in different ways, and you can use it as a template for manufacturing electronics, right?
Right. So folks have been working on this since, I've been hearing about it since about the mid-'90s, I guess, where some of the early work was just using a piece of DNA that had ends that were complementary to oligos that were immobilized on a surface. So then you put this piece of DNA down, and of course it sticks between the 2 oligos that you've got on the surface. And then you could coat it in something like gold nanoparticles. So now you've got a gold wire connecting these 2 places, and it's very thin gold wire. This is kind of an evolution of that concept, where they're using complementarity of DNA to build not only wires, but I gather they're building folded structures on the surface of the lithographic template. And a computer chip or any modern semiconductor is made with a photographic process. So they etch the thing on the surface photographically, and then I guess in this case they're building up with DNA additional features on top of that, and using the DNA as a method that they can control these very, very small, couple of nanometer features on the surface in order to build smaller, more compact chips.
Cool.
Yeah.
Well, I will post the link to that. That's very neat. He goes on to say, my question is, they are listing diarrhea as a symptom of swine flu. Wikipedia says diarrhea has been reported in some cases. Since influenza is an upper respiratory disease, or lower as well, and doesn't act in the stomach or intestines, how does it cause diarrhea? Is it secondary from swallowing phlegm, or is it just a case with so many people being infected that some percentage have diarrhea anyway? That's good. Those are 2 good ideas. So it doesn't replicate in the intestinal tract, influenza virus. And it's true that a number of the people with influenza are getting diarrhea. I've asked Peter Palazzi about this. He doesn't think it's because replication is occurring. It could be something else, a bacterial infection coinciding, or it could be a cytokine problem. You get a lot of cytokines produced, and it causes GI symptoms. But it's not likely from replication. This virus doesn't grow in the human intestinal tract. So we don't really know what's causing it so far, but those are some ideas.
It's also possible that it's spurious. Diarrhea occurs at a certain frequency, and people get flu at a certain frequency, so you'd expect some of them to have diarrhea.
Could be, right.
Or because they recommend a lot of fluids in treating flu, maybe that dilutes out the microflora of the large intestine, and you get loose bowel movements rather than frank diarrhea.
All right, we have 3 more. Let's get through them, and then we'll be happy that we did all our emails.
Yes.
Okay, Rich Condit. You know this fellow, Rodney. Why don't you read his email? And actually, this is up your alley. The next 2 emails have to do with our classification TWiV of a couple of weeks ago.
Okay.
And we made a couple of errors, so these are correcting. But that's fine. It's fine. Go ahead, Rich.
So Rodney writes, "Hello, I've been an avid listener of TWiV over the last 9 months or so and have very much enjoyed the podcast. It's always great to hear a broad, informed discussion on virology, and your recent podcast on viral classification was of particular interest to me. As a viral genome curator at the National Center for Biotechnology Information, NCBI, I'm currently working with the International Committee on the Taxonomy of Viruses, ICTV, and several virology research communities in an attempt to address long-standing and emerging issues of viral classification. Your podcast provided a concise primer to this topic, and I thought it was worth mentioning a few additional issues. First of all, viruses are classified by nucleic acid, dsDNA, SS, single-stranded DNA, double-stranded RNA, etc., both by NCBI and ICTV. Although these divisions are given no rank, they provide functionally relevant handles with which to globally group the viruses. And we can come back to that. Second, the way in which viruses are classified is somewhat variable. You might be surprised to know that despite developments in genomics, many viruses are still classified by morphology using electron microscopy. This is particularly true of the bacteriophages, and NCBI and ICTV are working hard together to develop more portable computer-based methodologies. However, it's worth noting that even when computational tools such as BLAST and PASC are used to classify viruses, the line of demarcation to separate different organisms vary widely among different families. This variability may actually be a good thing, as it allows different research communities to tailor taxonomy, making it more relevant to a specific group of viruses. Perhaps the more pressing issues in virus classification arises as the traditional isolate passage and physically characterized approach to viral discovery is altered by modern molecular techniques. This came up in the TWiV discussion. In the past, viruses have been characterized by a number of criteria, including host and disease, yet the proliferation of environmental sampling, emerging direct sequencing methodologies, and a greater appreciation of the broad host range of some viruses creates a number of classification problems. For example, how does one know if a novel candidate genome gathered from a sewer or a biopsy is actually a virus? And for that matter, how does one know that the sequence at hand is full-length, fully functional viral genome? The relevance of these questions grows every day as databases are filled with an increasing number of novel genomes obtained through these methodologies. Of course, these direct sequencing projects do have a number of advantages. There's no need to passage the prospective viruses over host cells, so there's no laboratory adaptation to a particular cell line. Such open-ended approaches also recover multiple isolates from a single sample, allowing one to track naturally occurring genetic diversity. And it's likely that the proliferation of new techniques combined with good old-fashioned bench work will fundamentally change our view of viral evolution and adaptation. Take care and keep up the good work, Rodney.
Wow, that was a fabulous letter.
Yeah.
I'm going to put this on the website.
That's fabulous.
Rodney is a graduate of our PhD program, I'm proud to say. Hello, Rodney.
Your chest is just butts at all of its buttons.
So,
did a good job.
That was great.
He did a really good job. So there's a couple of issues here. One is that in the early history of the ICTV, they wrestled with this question of, do we use a standard Linnaean classification system where it's monothetic. So each taxon is defined by a single characteristic, okay? And they decided against that because they couldn't decide what characteristics to use or which ones were more important than others, okay? Because you got to create a hierarchy. And I think that was a good decision. And so what they decided on was to use a polythetic approach. That is, a given taxon is defined by a collection of characteristics. Right. And Rodney points out a number of these, okay? Morphology, genome size, whether or not it's got an envelope, ecological niche, what the host is, a whole bunch of different things. And then they started to fill the taxonomy from the middle at genera. And then they defined species within genera and grouped genera into families. And importantly, not all families are grouped into orders. So order is the highest taxon that's used. There's 5 orders currently. That's in the next email. Not all families are in orders. So the family is the most general taxon to which any virus can be assigned. And so that's become the common reference point for referring to viruses, the family, because it's the most general description of a given virus. The grouping of double-stranded, single-stranded DNA, etc. ICTV calls that category the order of presentation. And I think that's because they group things into those categories and present them in that order whenever they publish their thing. And they apparently are reluctant to call that grouping a taxon, okay? That would be higher, whatever's higher than order ordinarily. So they're retaining that just as a grouping, an informal grouping. But interestingly, that even points out the polythetic characteristic, because one of those groupings, rather than being just nucleic acid, at fundamentally the same level, is whether or not it has a reverse transcriptase.
Yeah,
and that includes both RNA and DNA containing viruses. So it really has to be polythetic.
Yep.
The other thing is that the way they do this, they toyed with the idea of using a computer to handle all of this data in the polythetic approach, but it was just too complicated.
Wow.
So they decided to use committees. And so you get together a collection of scientists and let them hash it out.
The computer approach must have been really difficult.
Well, it speaks to the power of the human computer, if you like, because the fact is it works. I mean, I'm sure it's not very efficient, but it works. And so the summary quote that I heard by one of the ICTV dignitaries is that the system comprises the opinionated usage of data.
Right. That's great.
Well, I mean, you've got a square peg in a round hole problem here, because if you don't define viruses as being alive, then the Linnaean system is totally inapplicable anyway. It doesn't really matter.
Yep.
And in fact, your graduate student used the word organism there somewhere. I think I would strike that from his record, because...
He had it in quotes, though.
Oh, he did? Okay, that's fine. But, I mean... It's an intractable problem because of these multiple origins and multiple biologies that this thing is piggybacked onto, rather than being an origin itself.
And ultimately, I think now that I kind of mocked the idea, I think it actually does make a lot of sense to have humans doing this, because ultimately this is a matter of opinion.
Yeah.
The viruses don't recognize any taxonomy.
Right. So
that's right.
Why don't you do it like astronomy? They just number them. 1 through. And, they have new galactic catalog, and then they have a number after it, and you go to the coordinates and you get your telescope out, and bang, there's that star.
Well, we already got names. So
I know. It makes it convenient to be able to talk about them, but if you enter the plant virus world, they're less organized by far. I counted 1,400 sequences the last time I looked. Different viruses, 1,400 different plant viruses. That's wow. That's a lot of viruses.
Only what we know about.
Oh, that's right. Exactly.
I think we should point out that Rich authored the chapter in Fields Virology, the encyclopedia, basically, on classification. Is that right, Rich?
Yeah, a section of a chapter. I knew nothing about it when that happened. I was roped into writing a chapter called Principles of Virology, which is initially was supposed to be sort of lab techniques, plaque assays, that kind of stuff that are peculiar to virology, that I thought was basic information that you needed to know to understand how people collect the data for viruses.
Right.
And when I agreed to do that, they came back and said, "Oh, by the way, we're going to nuke the taxonomy chapter, so we want you to write an introduction to that. And we're going to nuke the genetics chapter, so we want you to do that." Genetics was okay, but taxonomy, I came in cold. So all you listeners out there, if I've got it wrong, let me know.
That's funny.
All right. Well, Rodney, in our next...
it doesn't sound like you can get it right, no matter what you do.
Rodney and our next writer, Elliot, will let us know. Elliot is he's a virologist, and he is involved also in classification. In fact, we had a TWiV some time ago with Chris Upton, and Elliot is a collaborator of Chris, and he's involved with classification. He writes, the International Committee on the Taxonomy of Viruses, and their website, provides access to a database of the current taxonomic classification of viruses, as well as the definitions and guidelines used by ICTV to make the classification. The taxonomic ranks used to classify viruses are order, family, subfamily, genus, species, as we've heard. There are 5 orders currently recognized: Codovirales, Herpes viralis, Mononegavirales, Nidovirales, and Picornavirales. All viruses are classified into species, including plant viruses, and investigators who study every recognized virus family are represented on the study groups that participate in the classification process, including plant virologists. So, as Rich said, it's done by committee. A viral species is defined by the ICTV is a polythetic class of viruses that constitute a replicating lineage and occupy a particular ecological niche. A polythenic class is one whose members have several properties in common, although they do not necessarily all share a single common defining property. In other words, the members of a virus species are defined collectively by a consensus group of properties. Virus species thus differ from the higher viral taxa, which are universal classes and as such are defined by properties that are necessary for membership. Note that poliovirus is no longer a species. It has been renamed human enterovirus C.
Really?
Yeah, and then polio is within that, of course.
And Pluto is no longer a planet.
It isn't. As for how different a virus isolate needs to be to create a new species, this came up, I think you asked this, Dick. This varies according to the virus family and varies especially according to the genome composition. DNA viruses vary much less than RNA viruses. Each ICTV study group provides species demarcation criteria specific for each virus family that define the criteria for establishment of a new species for that family. Okay, so it's different for each one, how much sequence different. Okay, so that's good because we didn't really know that.
Know, his that viral species definition actually encompasses an interesting potential definition of life. Replicating lineage that occupies a particular ecological niche.
Yeah.
And then you
just a thought.
Then you would then viruses would be living by that category. Well, they don't replicate on their own. But they do replicate.
They do replicate. And they establish a lineage and they occupy a niche.
So what are transposons? Are they alive?
I guess they would have to be, wouldn't they?
That's where I have problems with my own definition, because I want to think of viruses as alive, but I don't want to think of transposons as alive. So
it's a matter of opinion.
Yeah. That's right. Or prions. We can go into prions too if you're a proteomimus.
All right, Elliot ends up. Thanks for providing the opportunity for clarification. I think love the program. That's great. It's a nice way of saying you guys didn't get it right. Thanks, Elliot. We appreciate it. We provide a forum for information, right? We don't claim to know everything, but we like to talk about it. And finally, Chris wrote, to answer the question about NVIC, National Vaccine Information Center, someone asked me a few weeks ago if this was a good source of information about vaccines. He says, no, it is not a good source of information. Many of us have called it the National Vaccine Misinformation Corporation.
Dear.
While the information may have had a factual base at one point, it is not updated with new information. Also, a big red flag is their board includes a vaccine litigation lawyer, and there's more information about that organization and its founder in Arthur Allen's book, Vaccine. And he puts a web link to this information about the litigation lawyer. Okay, thanks, Chris. I didn't know very much about that NVIC. Good, we got through all the email. And how about some picks of the week?
Aha.
Rich, have you got a pick?
You bet. So I'm going to recommend a book called Stumbling on Happiness by a gentleman named Daniel Gilbert. He's a Harvard psychologist. And this is basically a book about how the brain works. It's a psychology book. It's nice because he describes psychology experiments. And so he gives you the methodology and the data to understand the conclusions that he describes. And there's a couple of really interesting things in the book. One of which I'll just give you an example is that memories apparently are not stored as the whole memory from start to finish. They're compressed. You store bits. All right. And when you recall it, you recall the bits and then you interpolate.
Right.
Okay. So that is an imperfect process. And he also talks about the imperfections in the process and how we frequently get stuff wrong. So it's very, very interesting psychological treatise on how the brain works.
Cool.
Very good.
Stumbling on Happiness.
Nice.
Yeah. All right. Dick.
I have a pick. It's not a book, it's a series of books, and I want the audience to be aware of the following connection between art and science. I recently loaned you my copy of Rat's Lice and History by Hans Sincer to Vince, and Vince obviously accepted it, and I said, do you know who Hans Sincer's technician was for 20 some odd years? And he said, I don't know what you're talking about. So I went to the bookshelf and I picked up a book by Elliot Porter, one of the great color photographers of our time. And I said, this guy was a technician in Hans Zinzer's laboratory at Harvard for at least 20 years and perhaps longer than that. But he was also a good personal friend of Stiglitz. And he liked taking photographs. And so all the time he was working in the lab with Hans, he was out photographing nature. And he would show his photographs to Stiglitz. And then one day Stiglitz says, you can quit your day job. He says, you now know how to take photographs and you should go out and photograph the world and record nature, because you're obviously interested in nature to begin with. So there's one of his books on Antarctica. Another one is on just a random sampling of nature shots that Elliot Porter has taken. He uses a large format field camera to take his photographs, just like Ansel Adams did, and except that it's color photographs. So it's really quite remarkable. I think he used film rather than glass plates, but still, to see the connection in his eyes between what he was studying in the lab and to go out in nature and find equivalents at almost all levels is just a remarkable connection, I think. And so that's what my recommendation is.
Yeah, I was just at Dick's office for lunch, and I saw this book by Hans Zinzer, and that's the origin of that story.
Right.
It's beautiful. Alan.
Yes, my pick of the week is a YouTube channel. It's YouTube USGOV-HHS. USGov HHS. This is the Department of Health and Human Services. I know everybody's yawning already, but wait. They have a YouTube channel, and on it right now they are hosting a contest to pick public service advertisements submitted by contestants. And one of the contestants, Dr. Clark, who I believe is based in New York, he's a physician. He does an H1N1 rap, which I thought was pretty darn good. But not that I'm a connoisseur of rap, but I thought it was a good humorous take that has the appropriate messages about hand hygiene and that sort of thing. 60-second.
I'm about to teach flu to the medical students. I'll give them that.
Sure.
Be interested in that.
In fact,
there are other contestants on the site too, and some of which are kind of clever.
When I was at TED, there was a guy that gave a rap on molecular biology that was absolutely fantastic. I forgot to mention that, but
Dick, do you want to make an entry for this YouTube contest?
No.
Okay. All right, my pick is a podcast which I've been listening to for quite a while, and I just realized this morning when I was listening that I hadn't picked it, and I like it. It's called the Coast to Coast Bio Podcast by two gentlemen, one on the East Coast and the other on the West Coast of the U.S., hence the name. And they talk about biology, programming, and everything in the middle. So these guys are slanted towards programming angles of approaching biology. But and for that, it's interesting to me because it's different. And they talk, for example, about programmatic approaches to literature and searching and storage and so forth, different programs that biologists use. They know biology, and they often talk about that as well. But I enjoy it. It's really good. So check that out, Coast to Coast Bio Podcast.
Neat.
And that will do it for another TWiV. Thanks, everyone, for joining. Alan can be found at dovedocs.com.
Yes.
And you can find his ramblings there. Thanks again, Alan.
Sure.
Rich, thanks for coming back.
Hey, great to be here. I really enjoy this.
We'll put your website link, and we will have you back next month. I understand you're all booked up for the rest of the month.
Yeah, the rest of the month is bad, but I'd love to come back again.
Is great. October okay for you?
Oh, yeah.
All right.
Find a day.
We'll find a day.
Cool.
We'll have you back. Dick Despommier, of course. Thank you very much.
Pleasure.
Go fishing.
Yeah.
Dick doesn't have a website. I'm just kidding.
Sure he does.
He's got many. He has a media empire. Which one should we mention? All of them?
Medicalecology.org I'd like them to look at now.
Okay.
That's one. There's a big section on infectious diseases, and they might enjoy it.
Okay. As always, send us your questions and comments to twiv@twiv.tv. I don't know if we'll have a show next week. I have a meeting I have a microbiology retreat to go to. So we may skip the week.
Or we could record it earlier.
Or we could record it earlier. So we'll work on that. And just in case the schedule doesn't work out, you'll know why you don't hear your weekly TWiV.
Oh, we have to do one, Vince.
All right, we'll do one.
Come on, you got me chafing at the bit here.
Very good. Thanks, everyone. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.